Next, we have the biochemically important monosaccharides. First is the D-Glyceraldehyde and Dihydroxyacetone. So, these are the simplest monosaccharide. No, sila yung pinaka-simplest. They contain 3 carbon atoms. No, they are trioses. And they are important intermediates in the process of glycolysis. You will be encountering these molecules sa finals, no, sa carbohydrate metabolism. Si D-glyceraldehyde and dihydroxyacetone. Si glyceraldehyde, siya ay, of course, aldehyde, and dihydroxyacetone, of course, ketone. And si dihydroxyacetone, it is a chiral molecule. Ay, wala siyang chiral center. Ito, walang chiral center. Same. Ito, naka-double bond. Ito, same. So, a chiral siya. While si glyceraldehyde, merong chiral center. No, isa lang ang kanyang chiral center since ito ay same na. Hydrogen. So, this is chiral molecule. Next is D-Glucose, ang pinaka-importanting monosaccharide. Okay? Called as grape sugar, dextrose, that is glucose, and blood sugar. And it is the most abundant in nature. It is the most important from a human nutritional standpoint since siya yung kailangan to produce energy. No? Cells use glucose as a primary source of energy. And the normal concentration of glucose in human blood is in the range of 70 to 100 mg per dl. So why do we need to fast 8 to 10 hours para ma-test yung glucose level sa blood? So kasi magiging falsely elevated yan kapag kakakain nyo lang, right? So, the actual glucose concentration in blood is dependent on the time that has elapsed since the last meal was taken. Kaya we need fasting, diba? Mataas yung concentration ng glucose sa blood kapag bagong kain. Mga 1 hour after eating. Kasi nga, nadadigest ng katawan and inaabsorb ng blood. So, ngayon, yung glucose nasa blood, hindi pa siya na transport sa mga cells, hindi pa siya pumapasok sa mga cells. Ayan, nasa blood pa lang siya. And the concentration decreases over the next 2 to 3 hours and back to the normal range. Kaya kailangan ng fasting. We call it hyperglycemia kapag high blood sugar. No, hyper, mataas. Glyce means sugar. Emia means in blood. Next, D-galactose, or we call it brain sugar. So it is synthesized from glucose in the mammary glands for use in lactose. It is a component of glycoproteins that are found in brain and nerve tissues, and also present in the chemical markers that distinguish various types of blood, known as ABO blood type. Now, the O type, the A antigen, and the B antigen. Again, it is an aldohexose, and ang differ lang ni galactose kay glucose doon sa orientation ng carbon 4 hydroxyl group. So, this is the carbon 4 hydroxyl group. 1, 2, 3, 4. So, doon lang sila nagkaiba. Next is D-Fructose, because we also call it Levulose or fruit sugar. So aqueous solutions of naturally occurring D-fructose rotate plain polarized light to the left, hence the name Levulose from levorotatory. It is the most important ketohexose, and it is found in many fruits, and it is the main sugar in semen. Itong si fructose and galactose, it can be converted into glucose, kaya nagagamit rin sila to produce energy. Next is D-Ribose. Again, si D-Ribose is an aldopentose. It is a component of a variety of complex molecules like RNA, DNA, and ATP molecules. So we have the ribose and deoxyribose. So ribose, OH sa carbon 2, deoxyribose sa carbon 2, walang oxygen. Kaya nga deoxyribose. And ang difference ni glucose sa ribose, wala lang yung pangatlong carbon. So this is removed. The rest, the same. No, kaya siya pentose. Next, ATP molecules. So, ito yung energy molecules natin. ATP stands for adenosine triphosphate. So, meron tayong adenosine, alam niyo na yan, diba? Sa nucleic acid. We have the adenine, a base. And then your ribose, ito yung adenosine. No? Combination ng adenine and ribose, adenosine. And then, triphosphate, tatlong phosphate group ang naka-attach. Kaya, ATP, no? Adenosine triphosphate. So, adenine, ribose, plus 3 phosphates. So, this is the ribose part. Now, let's go on to the Haworth projection. So, it is the cyclic structure, no, from open chain, from Fischer projection to Haworth projection, or the cyclic structure or closed form. It is invented by Norman Haworth. He is an English chemist. This will form cyclic hemiacetal. So, ito yung hemiacetal group part. So, paano ba mag-convert from open chain, no, from Fischer projection to Haworth projection? So, this is the step-by-step. So, we have here Fischer projection formula for the glucose, no, open chain. So, we have 6 carbons, right? And then, ito yung OH. That determines the D, glucose. Now, second, all OH, the hydroxyl groups to the right, itong mga nasa right side, appear below the ring, whereas OH groups to the left appear above the ring. So, itong nasa part na right, nasa baba yan ng ring, ito nasa taas ng ring. Okay, ayan. So, si carbon 2, yung OH na nasa right, so, dapat nasa baba siya. So, yung OH ni 3, carbon 3, nasa left, nasa taas. Si carbon 4, nasa right, so, nasa baba, except kay carbon 5. Nasa baba siya, dapat, pero meron tayong gagawin sa kanya, no? Special si carbon 5 na OH. Next, counterclockwise rotation of groups attached to carbon 5 gives this structure. So, magiging ganito na siya. Gulat kayo, no? Paano kaya yun? So, later, papakita ko sa inyo yung easiest way, no, kung paano ko siya i-convert. Then, next, the OH group on carbon 5, so ito yung carbon 5, adds cross the carbonyl. And then, itong H from the carbon 5, ilalagay dito, no, dito sa carbonyl group. Kaya siya magiging ganito. And meron pa tayong 2 stereoisomers possible, alpha and beta. So, bukod pa yung D and L. No, meron tayong alpha and beta. Alpha, kapag nasa baba ang OH sa carbon 1. Ay, yun yung palatandaan. Beta, kapag nasa taas yung OH. Yung H dito kay carbon 5, na-transfer dito sa taas, no? Dito sa OH sa taas, sa oxygen sa taas. And then, yung H dito, na-transfer dito sa baba, sa OH. Oxygen dito sa baba. So, again, Haworth Projection. Ito yung cyclic form. We have alpha glucose and beta glucose. Again, kapag alpha, hydroxyl group nasa baba, below, ay nasa baba, alpha. And then beta glucose, hydroxyl group nasa taas. So, beta. Now we have anomers. When we say anomers, these are cyclic. Okay? They are cyclic monosaccharide. No, cyclic monosaccharides that differ only in the positions of the substituent on the anomeric or in the hemiacetal carbon atom. And this is the hemiacetal carbon atom. Ito yung hemiacetal carbon atom. So again, we have the alpha anomer and the beta anomer. So again, ito, we have alpha D glucose, alpha. And one way para malaman kung D or L-glucose ang nakacyclic, titingin kayo sa carbon 6. This is the carbon 6. Kapag nasa taas si carbon 6, that is D. Kapag nasa baba, down si carbon 6, that is L. Again, kapag nasa taas si carbon 6, that is D. Kapag down si carbon 6, that is L. Clear? So, iba pa yung alpha and beta OH. So, ito yung mga kailangan yung tandaan sa positioning. Tapos yung OH, kapag right, down. Tapos yung OH, left, up. Next, cyclic forms of other monosaccharides. For example, we have the D-Fructose. This is the open chain of D-Fructose. So, convert natin siya into Haworth form. We have alpha D fructofuranose or the alpha D fructose. No, furanose means ito yung structure. And then pyranose kapag 6 atom ring. Kapag 5 atom ring, furanose, no, furan. Since ketone siya, ganito yung nangyari. So, we have carbon 6, carbon 5, and then yung OH separate. Yan. Magbaband yung OH dito sa carbon 2. And then, itong H, mapupunta dito sa carbonyl. Ito na yung OH. And then, same procedure. Ito, OH, nasa right. So, nasa baba. And then, carbon 3, nasa left yung OH, kaya nasa taas. So, again, pyranose, cyclic monosaccharide containing 6-atom ring. So, ito yung pyran, no? 6-atom ring. Pyranose kapag monosaccharides, no? Sugar. Kaya pyranose. And then, furanose, ayan, furan, kapag 5-atom ring. Now, ulit tayo. Paano nga ba i-convert si Haworth? Paano nga ba i-convert ang Fischer projection to Haworth? So, again, for example, we have glucose. So, para ba i-drawing si glucose sa Fischer projection? Ayan, so we have 1, 2, 3, 4, 5, 6. So, right, left, right, right. So, this is D-Glucose. So, paano natin siya mako-convert into Haworth? Sabi, counterclockwise. Okay? So, we have 1, 2, 3, 4, 5, 6 carbon. So, unang gagawin nyo, dito muna kayo sa carbon 6. So, dito is D-glucose. Diba? Para ma-identify kung D or L ang isang monosaccharide, nasaan si carbon 6? Kung D, glucose, up or down. Kapag D, nasa taas. So, ilalagay natin siya sa taas. Okay? Next is si carbon 5. Ito si carbon 5. Later na natin siya lagyan ng hydroxyl and hydrogen. Okay? Proceed muna tayo sa ibang carbons. Carbon 4, carbon 3, carbon 2, and si carbon 1 na merong hydroxyl bond at merong hydrogen. Okay? So, we have 1, 2, 3, 4, 5, 6. So, ayan yung carbons. Now, i-arrange natin yung mga hydroxyl group. So, carbon 2, ito si carbon 2, yung OH niya nasa right. So, sa baba ang OH. And then, nasa taas ang hydrogen. Next, nasa left ang hydroxyl group ng carbon 3. So, nasa taas, dapat yung hydroxyl. Next, so nasa right yung hydroxyl group ni carbon 4. So, nasa baba ang hydroxyl group. Okay? Now, dito na tayo sa carbon 5. Now, focus tayo dito sa carbon 5. Since nasa taas ni carbon 5, si carbon 6, automatic, nasa baba si hydrogen ng carbon 5. Ang OH ni carbon 5, andito. Okay? So, dito na yung magic na mangyayari. No, medyo confusing. Now, para maging close yan, para mag-close itong part na to, kailangan mawala itong double bond. So, para mawala itong double bond dito sa carbon 1, yung hydrogen dito sa carbon 5, dito sa hydroxyl ng carbon 5, idodonate dito sa carbonyl. No, matatransfer dito sa carbonyl. And then, ito, at is than, no? At is than. So, magkakaroon ng bonding ngayon dito sa carbon and oxygen dito. So, alisin na natin si hydrogen. For example, na-transfer na natin, mawawala na yung double bond dito. Magkakaroon na ng bond dito sa oxygen and carbon 1. So, dito ngayon, si OH. No? Since nasa baba ang OH, this is alpha D glucose. How about kapag nasa taas? So, OH and then H. Then, this is beta D glucose. Okay? Okay, so yun yung simplest way para maintindihan nyo yung conversion Fisher projection to Haworth projection. Now, let's go on to the reactions of monosaccharides. So, we have the following reactions. Redox reaction, no? Oxidation to produce acidic sugars, and then reduction to produce sugar alcohols. We have the glycoside formation, phosphate ester formation, and amino sugar formation. First is oxidation to produce acidic sugar. So, oxidation reaction. So, ito yun, yung nasa taas. Oxidation, no? Oxidation. So, obtained by oxidation, and we have 3 types of acidic sugars. Aldonic, aldaric, and alduronic acid. So, itong primary alcohol, kapag inoxidize mo, magiging aldehyde, and this aldehyde, pag inoxidize, will become carboxylic acid. So, for example, these are the acidic sugars, and it depends on the oxidizing agents that you will be using. First is the weak oxidizing agents. So, kapag weak oxidizing agents yung gamit nyo, it will produce aldonic acid. Aldo means aldehyde. Okay? So, for example, glucose, and gagamit kayo ng weak oxidizing agent, no? Example, ito is Tollens and Benedict solution. It will oxidize the aldehyde end, to give an aldonic acid. So, ito. Aldonic acid, no? Naging carboxylic acid, yung aldehyde. So, naging gluconic acid. No? Kapag weak oxidizing agent, yung aldehyde group magiging ma-oxidize to produce aldonic acid. Next, kapag strong oxidizing agent ang ginamit, no, it will produce aldaric acid. Kapag strong oxidizing agent ang gamit, both ends of a monosaccharide at the same time will oxidize. So, itong part na to, at yung terminal primary alcohol. No, yung carbonyl group at yung primary alcohol. So, magiging acidic, magiging carboxylic acid. So, this will become glucaric acid, no? Aldaric. Next, kapag enzymes yung ginamit nating agent. So, dito, yung primary alcohol lang, no? Will become carboxylic acid. And this will yield alduronic acid, or kapag glucose, it will become D-Glucuronic acid. Next, reduction to produce sugar alcohols. So, ito, no? Aldehyde to primary alcohols, reduction. So, we have alditol, no? Aldehyde and then all for alcohol. So, we have glucose, magiging glucitol. So, itong carbonyl part, ma-reduce alcohol, no? Add lang ng dalawang hydrogen to CH2OH. D-glucitol is also known as D-sorbitol. And these alcohols are used as moisturizing agents in foods and cosmetics. And also, it is used as a sweetening agent in chewing gums. Next is glycoside formation. When we say glycoside, this is the acetal formed from a cyclic monosaccharide by replacement of the hemi-acetal carbon hydroxyl group with an OR group. No reaction with an alcohol. So, we have here the methyl alcohol and your beta D glucose. So, this is the hemi-acetal carbon, right? So, ito yung hemi-acetal. So, i-replace lang natin yung nakalagay dito sa carbon 1. Okay? Alisin natin itong H, yung hydrogen, and itong methyl, mapupunta dito. So, yan siya. Magiging methyl beta D glucoside. Okay? Glucoside. And then, will produce water. Kasi yung water, yung hydrogen dito, mapupunta dito sa OH. So, meron tayong methyl alpha D glucoside kapag nasa baba, and then methyl beta kapag nasa taas. Next is phosphate ester formation. So, hydroxyl groups of a monosaccharide can react with inorganic oxyacid to form inorganic ester. So, an inorganic oxyacid is phosphate. Oh, we have phosphate ester. So, this is the structure of phosphate group. And ito yung structure ng phosphate ester. Now, pwede natin ilagay itong phosphate sa carbon 1 at sa carbon 6. Ito sa carbon 6. Kapag carbon 1, we call it alpha D glucose 1 phosphate kung saan naka-attach yung phosphate. And then, kapag glucose 6 phosphate, kung saan naka-attach yung phosphate, no? Nasa carbon 6. Kaya, glucose 6 phosphate. Next is amino sugar formation. One of the hydroxyl groups of a monosaccharide is replaced with an amino group. So, this amino group. So, usually, nasa carbon 2. No? Sa carbon 2. We call it, kapag glucose, glucosamine, and galactosamine, manosamine, no? And then, we have the N-acetyl amino sugar derivatives. So, meron tayong N-acetyl. Ito yung acetyl, no? Nakakonect dito sa nitrogen. So, we have N-acetyl alpha D glucosamine N-acetyl alpha D-galactosamine. So, later on, ma-encounter pa rin natin itong si N-acetyl amino sugar derivatives. Next, let's go on to disaccharides. Okay? So, tapos na tayo sa monosaccharides. Disaccharides na tayo. No? 2 monosaccharide units. So, carbohydrate that contains 2 monosaccharide units covalently bonded. So, through condensation, magbabind yung dalawang monosaccharides and through hydrolysis, mabe-break yung dalawang monosaccharide. So, disaccharides are crystalline, water-soluble substances. Example, we have sucrose, lactose, maltose. Next is the glycosidic linkage. So, si glycosidic linkage, it is a type of covalent bond. Okay? It is a bond in a disaccharide resulting from the reaction between the hemiacetal carbon atom of one monosaccharide and an hydroxyl group on the other monosaccharides. Dito sa isang monosaccharide, ito, yung hemiacetal carbon, and then hydrogen group of the other monosaccharides. Usually itong carbon 4. Depende. So, ayan yung glycosidic bond. So, again, kapag magbabind ka, condensation. No, maglo-loss tayo ng water para mag-bind. And then, hydrolysis kapag magbe-breakdown tayo. Kaya kailangan natin ng water to produce monosaccharide, no? 2 monosaccharides. Next types of glycosidic linkage, we have alpha glycosidic bond. So, alpha, nasa baba. And then, beta glycosidic bond, nasa taas. So, we have the following disaccharides that we're going to discuss. We have the maltose, cellulose, lactose, and sucrose. First is maltose. We call it malt sugar or maltobiose. So, formed from 2 units of glucose joined with an alpha 1,4 glycosidic bond. So, we have 2 glucose units. We have here the alpha D-glucose. So, alpha yung glycosidic bond, no? Alpha 1. So, ito yung alpha 1. Carbon 1. And then 4, nasa carbon 4. So, andoon yung bonding, no? Glycosidic bond. Kaya, alpha 1,4 glycosidic linkage. So, we have the maltose. Again, ito, pwede siyang alpha D-maltose kapag nasa baba, itong OH dito, and kapag nasa taas, and then beta D-maltose. So, maltose can be broken down to glucose by the enzyme maltase, which catalyzes the hydrolysis of the glycosidic bond. Next is cellulose. It is a reducing sugar, consists of 2 beta-glucose molecules linked by a beta 1,4 glycosidic bond. So, dito, beta D-glucose, kaya beta 1,4. So, ayan, taas. Then, 4. So, pag ganyan siya. Or pataas. Beta 1,4, no? D-cellulose. Next is lactose or milk sugar. So, it composed of galactose and glucose subunits. It makes up around 2 to 8% of milk. So, we have the bond beta 1,4 glycosidic linkage. So, si beta D-galactose, ito, and si D-glucose sa 4. Okay? Kaya, beta 1,4 glycosidic linkage. So, lactose intolerance. kapag lactose intolerance, it is a condition in which people lack the enzyme lactase. No, si lactase, siya yung enzyme na nagka-catalyze ng hydrolysis of lactose, no, para makabuo tayo ng glucose and galactose. So, kailangan siya for digestion ng lactose. No, we call it also lactase persistence, no, lack of lactase enzyme. So, kapag meron kang lactose intolerance, konti lang yung lactase mo, konti lang yung enzyme mo, konti lang yung lactase mo. So, yung mga lactose, for example, hindi siya nadadigest completely since kulang ka sa enzyme na lactase. So, hindi siya nadadigest, hindi siya nagpo-form into glucose and galactose completely. So, may mga lactose na hindi nadadigest, hindi na-absorb, nagre-remain sa ating intestine. And these lactose molecules will attract water, and that's the cause of discomfort, no, diarrhea, feeling fullness. And yung mga bacteria sa intestine, for example, E. coli. E. coli is a lactose fermenter bacteria. So, ginagamit niya yung lactose, produce lactic acid and gas, no? Adding to the discomfort. So, kaya yung iba, dumudumi agad matapos uminom ng mga dairy products. Next is sucrose or table sugar. It is the most abundant of all disaccharides. The molecule composed of two monosaccharides, which are the glucose and fructose. And the bond here, we have the alpha 1 beta 2 glycosidic linkage. So we have alpha glucose, no, si alpha 1 glucose sa carbon 1 ng glucose, and sa carbon 2 beta fructose. Ito yung beta, nasa taas yung OH, carbon 2. So pag ganyan yung linkage, alpha 1 beta 2 glycosidic linkage. And sucrose is not a reducing sugar kasi nawala na yung hemi-acetal group dito. Hindi na siya makakapag-react, no? Hindi na siya makakapag-reduce. Kaya it is not, or it is a non-reducing sugar. Next is oligosaccharides. Carbohydrates that contains 3 to 10 monosaccharide units covalently. So, complete hydrolysis of an oligosaccharide produces, of course, monosaccharides. So, we have two naturally occurring oligosaccharides that are found in onions, cabbage, broccolis, no, usually mga beans. So, these are the raffinose. Raffinose contains 3 monosaccharides, no? Siya ay trisaccharides. We have galactose, glucose, and fructose. Kompleto siya. Raffinose. And stachyose. Stachyose contains 4 monosaccharides. 2 galactose and glucose and fructose, no? Stachyose. So, ito yung mga linkages na present sa kanila. Okay? Next is polysaccharides, no? Polysaccharides or glycan, no? It is the alternative name for polysaccharide. Glycan. So, polysaccharide, a polymeric carbohydrate that contains many monosaccharide units that are covalently bonded to each other. So it contains several thousand of monosaccharide units. So example, we have cellulose, starch, glycogen, heparin, hyaluronic acid. So we have different classifications also of polysaccharides, no? Based kapag structural or storage for storage, and then acidic polysaccharides. And then yung homopolysaccharide and heteropolysaccharide. So kapag sinabi nating homopolysaccharide, same lahat yung monosaccharide units na meron sa kanya. For example, lahat yan glucose units. So, it is homopolysaccharides. Kapag hetero, of course, iba-iba. No, may glucose, may galactose, may fructose. Next, important parameters that distinguish various polysaccharides from each other. So, we have the following. First is identity of the monosaccharide repeating units in the polymer chain. So, yun nga. Kung homopolysaccharide ba siya, kung puro isa lang, for example, puro lahat glucose, and then heteropolysaccharide, no? Iba-iba. And then, next is yung length of the polymer chain. Kung short, kung long, and then type of glycosidic linkage between monomer units. So, we have alpha 1,4, alpha 1,6, and so on. And then, degree of branching of the polymer chain. So, meron tayong, dito, meron tayong unbranched and branched. Meron ding highly branched and slightly branched. No, less branching. So, dito tayo sa storage polysaccharide. So, polysaccharide, that is a storage form for monosaccharides and is used as energy source in cells. So, lahat ng i-discuss natin dito sa storage polysaccharides are homopolysaccharide. Okay? So, it only contains glucose molecules. So, number 1, we have glycogen. So, si glycogen sa yung storage form of glucose. So, it is multi-branch polysaccharides of glucose that serves as a form of energy storage in humans and animals. It represents the main storage form of glucose in the body again, and it is found in liver cells and muscle cells, the storage sites for glycogen in humans, liver cells and muscle cells. And we have two glycosidic bonds present in glycogen. First is alpha 1,4 glycosidic bond, and we have alpha 1,6 glycosidic bond. So again, meron tayong two types ng glycosidic bonding ng glycosidic linkage. First is yung alpha 1,4. Ito yung alpha 1,4. Ayan yung alpha 1,4. Si alpha 1,4 glycosidic bond, siyang responsible for linear, no? Yung paganyan na bonding. While si alpha 1,6, ito si alpha 1,6, alpha 1,6 glycosidic bond, siya yung branch point, no? Sa kanya, nagkakaroon ng branching sa alpha 1,6 glycosidic bond. So again, glycogen is highly or multi-branched, no? For every 10 glucose units, nagkakaroon ng branch. Next, let's go on to starch or amylum. It is polymeric carbohydrate consisting of large number of glucose units joined by glycosidic bonds, and it is produced by most green plants as energy storage. Kaya most of the food that we eat is starch, no? Kasi sa plants siya nakukuha. Sa plant siya present. We have two types of starch, the amylose and amylopectin. Amylose is unbranched starch. No, it is helical in structure. Kaya ang present na bonding lang sa kanya ay alpha 1,4 glycosidic bond for linear branching, 'di ba? Kaya nga siya unbranched kasi si alpha 1,4 lang yung present sa kanya. While si amylopectin, it is the branched starch. So meron siyang alpha 1,6 glycosidic bond for branching. So, 'di ba, siya yung branching point, si alpha 1,6. And then meron pa rin tayong alpha 1,4 glycosidic bond, same as the glycogen. Ang pinagkaiba lang nila, it is less, no? Less yung branching point. Less yung branching niya compared to glycogen na multi-branched. So again, starch, we have amylose and amylopectin. Alpha 1,4 for linear bonding, and then alpha 1,6 for branching point. Next is structural polysaccharide. So this serves as structural element in plant cell walls and animal exoskeletons. Under structural polysaccharides, we have cellulose, no, structural component of plant cell walls. This is the most abundant naturally occurring polysaccharide, and ito yung mga woody portions ng mga plants like stem, trunks. And it is linear structure, no? Fibers, filaments. So we have beta 1,4 glycosidic bond here, no? Ito, beta naman dito ngayon. Pero linear pa din yan. Okay, so ayan. Yung mga fibers. And then we have chitin. It is a primary component of cell walls in fungi, the exoskeleton of arthropods, such as crustaceans like crabs, lobsters, shrimps, and insects. And here we have the beta 1,4 glycosidic bond. So, ang difference ni chitin sa cellulose, may present siya ng N-acetyl D-glucosamine. No, they are N-acetyl glucosamine. May presence ng nitrogen. While si cellulose, wala. So, dito sa chitin, yung strength ng polymer matrix is higher due to the increased hydrogen bonding capacity. Kaya matigas, 'di ba? Yung crab, yung shell. Usually sa shell 'to makikita. No, yung chitin. Next is chitosan. Si chitosan, it is a derivative of chitin. Tinanggal lang natin yung N-acetyl, kaya N-deacetylated derivative of chitin. So, diba ito, yung chitin, mawawala lang tong acetyl part. Magiging ganyan na siya. Yan yung chitosan. So, yung chitin, i-deacetylated natin to produce chitosan. And si chitosan, marami siyang use, no, for biomedical devices, drug delivery, catalyst, water purification, for cosmetic. And again, for structural polysaccharides, we have chitin, chitosan, and next is acidic polysaccharide. A polysaccharide with a disaccharide repeating unit in which one of the disaccharide component is an amino sugar and one or both disaccharide component has a negative charge due to a sulfate group or a carboxyl group. So, dito sa acidic polysaccharide, they are heteropolysaccharides. So, iba-iba yung monosaccharide units. It involves in variety of cellular functions and tissue. We have two here, the hyaluronic acid and heparin. First is the hyaluronic acid or hyaluronan. Hyalus means glass. Hyaluronic acid solution have a glass-like appearance and it is highly viscous. No, it serves as lubricants. No, jelly-like consistency. So it is a linear polysaccharide. We have the N-acetyl beta D-glucosamine or NAG, and we have D-glucuronate repeating units. So it is linked by alternating beta 1,4 and beta 1,3 glycosidic bonds. So we have here beta 1,4 from the NAG and the beta 1,3 dito sa glucuronate. Next is heparin. It is an anticoagulant. No, it helps to prevent blood clots. So sulfate derivative of the glucuronate and a doubly sulfated derivative of the glucosamine repeating unit. So ito siya. We have the alpha 1,4 glycosidic linkage in heparin. Next, we have the glycolipids and the glycoproteins. Glycolipids, these are lipid molecules that has one or more carbohydrate units covalently bonded to it. While glycoprotein, of course, a protein molecule that has one or more carbohydrates. So glycolipids and glycoproteins, they are cell recognition, and this generally involves the interaction between the carbohydrate marker of one cell and a protein embedded into the cell membrane of another cell. So para ma-identify nila, no? Ma-recognize nila yung isang molecule or yung isang cell. Next is dietary considerations and carbohydrates. We call simple carbohydrate if it is dietary monosaccharide or disaccharide. Simple, okay? And then complex kapag polysaccharides na, dietary polysaccharides. Now, we have the natural sugar and refined sugar. Natural sugar, these are the sugars naturally present in whole foods like mga fruits. And then refined sugar, sugars that have been separated from its plant source. So like yung ating mga table sugar. No, na-extract na, kaya refined, no? Yung mismong carbohydrate lang, yung sugar part. Unlike dito sa natural sugar, meron pang ibang components, of course, yan. No, katulad nito, natural versus refined sugar, we have the orange versus the table sugar. So, alin yung mas healthy? Of course, yung natural sugar. Diba? Kasi, dito sa orange, hindi lang naman sugar yung meron kay orange. Meron pang other nutrients na like vitamins that will help you in converting carbohydrates into energy. While dito sa refined sugar, of course, sugar lang yan. No? Yan lang yung monosaccharides lang yung meron dyan. Yun lang yung mapakinabangan natin dyan. Kaya nakaka-diabetes yung mga refined sugars. Mga soft drinks, matatamis. Yung mga refined sugars kasi yung concentration ng sugar, ng monosaccharides, ng glucose, dumadami. Sobra-sobra. Minsan nag-accumulate na sila sa blood. Unlike sa natural sugar, marami pang nutrients ang meron sa kanya. So, that's all for this screencast. I hope you learned something new.